The overall goals of the following experiments are to determine the effects of NTBC on pyomelanin production and sensitivity to oxidative stress, as well as antibiotic minimum inhibitory concentrations, or MICs. This is achieved by adding NTBC to bacterial cultures to determine the concentration that reduces pyomelanin production. As a second step, bacterial cultures are treated with NTBC and antibiotics, which will allow the user to determine if NTBC treatment has an effect on antibiotic MICs. Next, NTBC-treated cultures are exposed to hydrogen peroxide in order to determine sensitivity to oxidative stress with NTBC treatment. Results are obtained that show treating pyomelanogenic bacteria with NTBC leads to a reduction in pigment production and increases sensitivity to oxidative stress while having no effect on antibiotic MICs, based on oxidative stress response spot plates and microtiter MIC assays, respectively.
The main advantage of the oxidative stress spot plate technique over existing methods, like viable counts and disk diffusion assays, is that lower concentrations of hydrogen peroxide can be used, rather than the high concentrations of hydrogen peroxide used in other assays. Generally, individuals new to our method of determining antibiotic MICs will struggle because a lack of precision in pipetting technique introduces variability between replicates. This variability can make it difficult to accurately determine the antibiotic MIC.
One day prior to the MIC assay, prepare cultures of the strains to be tested in LB with and without 2,2-nitro-4-trifluoromethylbenzoyl